Recording going. Okay. The styloid process was that pointed tooth or fang-like appendage hanging down from the skull in front of the mastoid process. It is labeled in one of our lab diagrams. Okay, and you showed it to me yesterday. The styloid process is one of the questions, and it is one of the answers on the test on Tuesday. Okay, I'll email you this list. Okay, I'm not making a secret of it. Anything I tell you about, you know darn well it's going to be on the test. Okay, you know where the vertebral foramen is. The vertebral foramen, you could stick your little finger into it, say in a thoracic vertebra. Okay. As you go around the circumference of the vertebral foramen, you're touching the edge, and that's the arch. That's the vertebral arch. Okay. I will probably be able to, I know I won't be able to show it to you today on real bones. Okay. And I'll send you a quick email with vertebral arch in it, in case you forget or didn't see what we did because you're out of the room or something. You can easily look it up on Google or in the book. Vertebral arch is one of the answers on the exam. Okay. I didn't tell you this, but I have been doing it to you or with you. I've been showing you bones and asking you, is this a right bone or a left bone? Is this the anterior or is the posterior? Didn't do it on every bone, but I did it on things like the femur and the humerus and the tibia, okay, whether they're right or left. And there will be about 3 questions on the test that show you a bone and ask you, is this right or is it left? Okay. So as you look at the bones, you know, pay attention to their orientation. There'll also be some x-rays. The x-rays number around 4, and not many, obviously. It's not the bulk of the test. Only about 4 x-rays. And believe me, these x-rays could be, you know, masterpiece drawings. They are beautiful x-rays. And you should have no trouble with it if you would not have had trouble with the bone itself. Okay. On Tuesday, the only thing you'll do that day is the practical. Okay. And I'm sure some of you will email me, you sure I missed this one or that one? It happens all the time. You will not be the first person to do that, okay? I've gone through the exam, and I did find that there's one duplicate question, okay? It's not a tough question. It was there on the last time I gave this exam, and I never realized it. And no one in the class brought it to my attention because it was a duplicate of probably one of the easiest things in the world. So I'm going to leave the duplicate question in there for you. Okay. Get it right twice. Okay. And you'll notice which one it is, I'm sure. And if not, I'll tell you later. But I'm not worried. I think that it actually helps you.
And
now,
Charlie, oh, sorry.
Go ahead.
When because I know when I did my last biology class, when you got into it, it asked for passwords to get into the test. That you don't have anything that we need to know for like passwords or anything, right?
No,
Okay.
I only use passwords when I'm giving a test late, early, at some other time for one student or something out of necessity. Then I assign them a password because otherwise, all the rest of you will stumble upon the test and find that it's open, and of course, then I don't have a test. Okay, but no, there's no password. You just go straight into the test from the module. Okay, that'll be explained again to you over the weekend when I send that other email. Now, this is important, I think. Here you are, and Valeria and Ashley, I don't have video for you today. Uh, thank you. Okay, that was what gets you 1 point of extra credit. Being here, participating a bit. We're doing a little bit more participating as we get close to the test. Everybody gets anxious. It really got to calm down. Okay, how many points of extra credit can you get on the practicum? It depends on how many days you were here. For every day you were here and your video was operating for at least half of it, you don't have to do it for all of it, then you'll get 1 point. We have first week, two opportunities, second week, 2 opportunities, and this week, and counting today, 2 opportunities. That's 6. A couple of people weren't here the first day of class, okay, because I added them late. Well, their bonus was to be added, I hope, but they will not get extra credit for something they did not do. But it's only 1 point, and that's not important at this juncture. Okay, so the most that you could get is 6. And how do I keep track of that? It's kept track automatically in ConferZoom. I go in, compare the length of time you were in there with us every day, and then I mark it down in my role. Okay. If you later on say, "Oh, I was there that day," well, I'm going to stand by ConferZoom, and I'll show you the page if I need to. And if your name's not there, you don't get the credit. Don't worry. Canvas and ConferZoom do make occasional glitches, but I have not found any glitches in this, the attendance. Okay. Now, I need 4 outlines from you. Some of you have actually got them all done. Others are working on it. Don't wait much longer because you back yourself right up to the exam. Need chapter 1, 6, 9, and 2. Okay. If unfortunately you don't have all 4 of them in before 2 o'clock when the test starts, then you forfeit all that extra credit. You forfeit the ConferZoom extra credit that I'm giving you right now. Okay. I think it's important to read those chapters. I think it's more important than you do. When I was a student, I would have felt the same way. Okay. So I need to have those, and don't send it after the exam. Don't tell me I forgot to send it. Hear that one a lot. 2 o'clock, I'll send you a reminder, okay? And please, please, do yourself a favor and don't wait till the last minute. Any questions about that?
I have a question.
Yes.
Do we have to have our cameras on for the practical or for the test?
No.
No.
You're taking the practical exam in your own space, okay? You're not taking it in Zoom. You're not taking it in ConferZoom. You're taking it off of the page.
Okay, because my Zoom doesn't work on Chrome for some reason.
Pardon me?
My camera doesn't work on Chrome. That's why I always use Safari. You know what I mean?
Okay, well, you're fortunate. I see what you're saying. And it's good that you're not going to have to use your camera, because you're going to have to use Chrome.
Okay.
If you use Safari, let me tell you right now, if you use Safari, you're going to crash. Not just you, I mean, all over the place. Safari doesn't work well. It'll work for a while, and then halfway through the test, you're stuck. It's frozen. The rest of the pictures don't come up. So let's eliminate all the possibilities of these things happening. When they do happen, you email me, and here I am in the middle of the exam helping other people too. And now I got to solve that problem, if it can be solved. Sometimes I can't solve it. Well, actually, I believe that some time will come when I can't solve it. So far, I've always been able to solve the problem. Problem comes down to starting the page. Okay, you know how to set the page? Most of you, of course, do. Some of you have the F5 button on your computer. Now, don't do that in the middle of a test unless you get stalled. But at the top, and I'll show it to you when we get to Zoom in a moment, or share. At the top left, usually, there's a arrow in the form of a circle. Okay, that's reload. And you can hit reload, and a lot of times you can get that test going again. Okay. Sometimes I've had it where somebody, again, using Safari or something else, Explorer or whatever, can't get the test going again. So I have them submit the test in an incomplete state and start over again with the proper browser, and I get load up permission for them to take the test one more time, and then they start again. There's always a way around it. Just don't like to do those things. Okay. What else?
So does that mean on Tuesday we don't have to log into Zoom at all?
That's right. On Tuesday, come for the practicum. You log into Canvas. You go to the test. It'll be the bottom of the third module or the top of the fourth one. I'll tell you where. You have no problem finding it. And at 2 o'clock, it will open for you. Okay. And I'm giving you an hour and 20 minutes. That's longer than it really should. That's what we give face to face in the classroom. In the classroom, the students have to wander from desk to desk, and they have to wait for somebody else to get their butt up out of the seat so that they can get in there and answer the question. Okay? And so sometimes they're lined up for certain questions, and this is just gobbling their time up. Okay? Now, on future exams, I may not give you an hour and 20 minutes. Why? Well, for one reason, I just explained, you don't need as much time as an in-class person does. Not really. And to be honest, okay, I don't want to give anyone too much time to be able to look up answers. Okay, then not having the camera on you. Okay, I don't want that. Okay. But if I feel people are looking up the answers and such, then I will ask them individually to put their camera on. Okay, so let's just leave that little topic for the moment. Okay.
Charlie, you said that we can see how we did right away at the end,
Yes,
like when we submit it. Okay,
yep.
awesome. Thank you.
It should show up right away. Maybe it may take 30 seconds or something like that. I don't know, but
I can wait that long.
now you finish the test, you see it, you want to ask a question, you're going to wait. I got the rest of the class still, and I'm waiting for them to ask questions about the test. So I'm not interested in the one question that you missed that we can solve later on. Okay. As I said, I think the test is, except for the double question, I think the test is perfect this time, and I don't think we're going to have any problems with the exam itself and the questions and answers. Okay.
I have a question. One more
Yep.
question. Can we? So if we like don't know an answer right away, can you go back to this?
Yes.
Yeah, you can like fill out the ones you do know and then go back.
It's possible to set the test to go one question at a time and never go back. I don't do that. You can go back and forth any amount of times you want. If you don't know the question or the answer, flag it. Okay, there's a little flag there for you so that you can find it again later. Okay, all of this activity that you're doing, you're on question one. Student has viewed question one. Student has answered question one. Student has flagged question two. Student has gone on to question three, and then returned to question two. Everything that you're doing is being recorded, okay, in text. And the main thing that you cannot do, you cannot leave the test. By leaving the test, I mean you, like on a computer, you change from one window to the next. You got to stay on the test window. One or two times you might stray, you might hit your arrow on the side or on something that causes it to move. I don't care one or two times, but I've had people on a 100-point test leave the test window 110 times. They're looking up answers. They're looking up answers, and I just tossed their test. Okay, tell them why. Okay, so that's the fairness of it, I guess. What else you got?
I have a question on the in the modules on one of the labs. There's the foot labeled, and then there's the two that are unlabeled, like dashes going off. I'm assuming those don't need to be named.
Is it on the module you said?
Yeah.
Okay, we'll get there in just a second, and I'll let you
Okay.
show it to me because I'm not understanding what we're talking about. Okay, and you show me, Billy. Okay, when we get there. Okay. Well, I think we're getting more discussion of one sort or another right here than we will out of the discussion section. There will be a discussion section next week. It'll be on Thursday of next week. I'm going to rearrange the order of the class, but not the time. I'll start the discussion at 2 o'clock, and I'll start the class at 3. Okay, it'll work out the same way. Okay. But that's after the practicum, and the practicum is the next thing, and that's the one we got to concentrate on. Okay. Anything else?
Okay, I'm so sorry. I have one last question. For Bio 110, it doesn't show up on my Canvas.
Right, I wrote you.
You did?
Uh-huh.
Okay, I'll look at it later.
Okay. You know, if you write me, I write you. Okay, every time. Once in a while, if I'm out someplace, I don't know, it may take me a couple hours. Usually, I'll get right back to you. Anything else? Okay, let's move on. Okay, Billy, where am I supposed to look to see what your problem is?
I don't remember. It's whichever one has the foot in it. And I think it's just they're just considered like metatarsals, whatever 1 through 5. But there's lines coming off, and I'm just making sure it's not anything
Oh,
more
okay,
specific. Because I'm pretty sure it's just like metatarsals, but those lines just I was just making sure.
okay. Here?
Yeah.
Yeah, okay. Lines that don't have anything. This one is metatarsal 1, okay? This is just showing the proximal end of it. I took out the lettering. Yeah, I took out the lettering myself. When you got a PDF, it's kind of hard to take the lettering out. So what I did, I made a white background and overlaid it. So now you can't see the word that's there. Okay, I should have left that one, metatarsal 1. Okay, and the one in the middle here, this is a proximal phalanx. We don't need the ends. Okay, if I'm going to ask it, it's labeled. It's labeled. Now, the metatarsal 1 would probably be labeled someplace else.
Charlie,
Yeah,
I have a question. The knee is on the test,
the what? Knee?
knee diagram.
Yes. And I need to do that now.
Okay.
Okay.
Thank you.
Actually, I'm going to do that now. I don't need to do it now because it's really part of lab. But let's get this knee out of the way. Sorry. That's what I wanted to do. Okay, this is the same drawing that was there the other day. I told you I would review it because I was disappointed with something. And it turns out that I've been left behind. It looks like the world of anatomy is switching names again on one item, okay? And it's properly named here, but not on my study guide. So let's get to it. On this side over here, on the right side of the picture, this is the right leg. Here's the fibula. The fibula is always lateral. We know this is not the back of the well, it doesn't matter. The fibula is always lateral. The fibula is joined to the condyle on the lateral side of the femur. This is the lateral condyle here. And this is a ligament. It's called the lateral collateral ligament. I like that term. Lateral collateral ligament. Lateral because it's strictly on the lateral side. Collateral because there's going to be one on the other side over here. And a ligament. That's the proper use of the term ligament. When connective tissue joins bone to bone, that's a ligament. This is joining the fibula and the femur, bone to bone. On the other side, we have a longer ligament joining the tibia down here to the medial condyle of the femur. How do I know that's the medial side? Because the fibula is always lateral. Got to get that down. That's the kind of thing that if I went into a class, I'd take a pen and ink and write it on my hand. Fibula is always lateral. Okay, so we have a ligament. This is the medial collateral ligament. And it's a true ligament because it joins bone to bone. On top of the tibia are two rounded troughs. They're called meniscus singularly, menisci plural. On this side, this trough is the lateral meniscus. This is the medial meniscus. And the condyles of the femur fit into them and rotate. Lateral meniscus, how do I know? It's on the lateral side with the fibula. The medial is always on the other side. Let's come over here. Here's the patella on where we're leading. But here's the patella with connective tissue. Above the patella and joining the patella to four muscles called the quadriceps. We're going into muscles next Thursday. Quadriceps, vastus lateralis, you don't need to know the names, vastus medialis, rectus femoris, and vastus intermedius. There's four muscles. Slap the top of your thigh and then spread your hands out while they're on top of the thigh, and you'll probably be able to grab or touch all four, three of the muscles that I mentioned. The fourth is buried deep. That's the intermedius. That's the muscles on the front of the thigh. This is the quadriceps tendon. A tendon joins properly bone to muscle, not bone to bone. So to call that the quadriceps tendon is perfectly correct. On the inferior part of the patella, connective tissue joins it to the tibial tuberosity. Now here's where I had my problem. The diagram is calling it the patellar tendon. My study guide calls it the patellar ligament. Why? Because it's joining bone to bone. Many of the books still say patellar ligament, but I guess I missed the transition to wanting to call this patellar tendon. Okay. On the exam, I'm sure this is one of my favorite questions. I'll have it as a potential answer, and I would do this for you. I would say patellar tendon, parentheses, patellar ligament, so that you don't make any mistakes. When I put something in parentheses, one after another, it means they're both equal. Okay. I'll probably continue to call this patellar ligament for a couple of years and find one of the few remaining images that show it that way too. Now, here's the fibula. Here's the lateral collateral ligament. It's also known as the fibular collateral ligament if you want to confuse yourself. Lateral collateral is easier. Here's the medial collateral ligament. Pull the patella away so that you can see and hear. And you see another ligament going across. That's the anterior cruciate ligament. On the posterior side behind it, forming an X, is the posterior cruciate ligament. Over here, they've removed one ligament to show the posterior cruciate ligament here, and the anterior cruciate ligament unlabeled. You see it dark in the other side. Now, if you've ever watched football, especially, they get themselves some jock who used to be a player. They get him out there to do the sports announcing. And usually there's two of them, they're talking back and forth about stuff we don't even know. But it won't be 10 seconds before they spit out the term ACL or PCL, the anterior cruciate ligament or the posterior cruciate ligament. Anterior, right under the patella, posterior behind that. You may abbreviate or find that I abbreviate on the test, ACL, and I also use PCL rather than write it out. Those are the only two that I'll abbreviate. I won't abbreviate the collateral ligaments.
Charlie,
Yes.
can you injure one without the other?
Can I do what?
Can you injure the anterior and not the posterior? Or if you injure one, the other one is injured as well? Do you know?
No, you can injure them independently.
Separately? Because I hear a lot of ACL, but I don't hear a lot
Yeah.
as much.
I don't remember where I heard it this weekend. One guy's in for his third ACL operation. Jeez. Well, I had my on the medial side right here. I had this part removed, and then they stuck a plate in there, Teflon or something, and they put some coating over this, which they shaped, and called it a partial knee replacement. I had that done last December. It was the most painless thing I can remember. Absolutely no pain whatsoever. I never needed an aspirin even, although they gave me a whole bunch of oxycodone pills. They just sit over here as a reminder. So if you've got that ever in your future to look forward to, here's one person to tell you, go for it. I hobbled around for a year, not being able to walk very well, and I was so used to hiking all my life. Now I got the ability back.
That's amazing. I've not heard that before. That's awesome.
I actually, when I go around, especially to a hospital, why for myself, no, the doctor might be in there. Also to visit other people and things. I always run into somebody that's been a student of mine. I remember once around the first examination time, just like you're taking right now. It was in early October, and I'm sitting at home with chest pains by myself, living alone. And I called up a friend and mentioned it and everything. Said, let me know when you need to go to the doctor, when you need to go to the emergency room. I'll take you. I'll take you. No, I don't want to go. It took me 4 or 5 hours to want to go. That pain just kept on going. And so I got to the hospital, get up to the emergency room, and they check you over real quick. Anytime you have chest pain, you go right to the head of the line. And here's this guy taking my blood pressure, and he said, "Charlie, how you doing?" It's one of my students. I couldn't believe it. Oh God. Okay. So then they take me into the back room and give me an injection that will stop any pain from the digestive system, stomach pain or anything like that, so that they don't get confused. And it didn't have any effect. That means that's a heart attack. Okay. Well, this was when I was 47, and they decided they wanted to do a chest X-ray to be sure nothing was broken there, just to be sure. So they put me in a wheelchair, and they're pushing me along. Here we go into the X-ray room for a heart attack. Huh, weird. And I hear this thumping sound. Somebody running. Somebody big is running on that linoleum type floor. And it goes, Charlie, is that you? Is that you? And I said, yeah, it's me. He said, how did I do on my test today? How did I do on my test? That's all I cared about. I couldn't give a damn about his test at that point. When they wheeled me out 3 days later, I heard the same thumping. How did I do on my test? Did you grade my test? Over and over again. And I'm sorry to take up the time, but since I'm on this topic, I'll never get back to it again. I was visiting a person at Mission who had lost their eyesight, sudden loss of eyesight, and they had to get steroid treatment immediately, tons and tons of steroid, and that can bring it back. In fact, in this case, it did bring it back. But I'm in the emergency room in a special area that they've got for people like that. And I go up to visit real quick, go up to the front desk. It's a student of mine. She's the head nurse. She wants to talk. No, not now. Okay. I get in the room and they're pushing another patient outside of the hallway, and it's glass between the two. They're pushing this patient, a woman. She's lying down, and she's on a gurney. And the woman doing the pushing, the nurse, she sees me and goes, "Hi, Charlie." And then the patient sits up. "Hi, Charlie." God, they're everywhere. They're everywhere. Okay, now we need to back off. Cell division. Okay, this is the last of the cell type topics that we're going to have to do. Then we can get right into our test, and after that, into the muscles and the renal systems, as I call it. Okay, there are two different types of cell division, and I know you've had this before, but I know that we also get to where we are on the basis of what we once knew and now have forgotten. The two types of cell division are mitosis and meiosis. Meiosis is sometimes called somatic cell division. Somatic meaning of the body, whereas meiosis is referred to as reduction division and only occurs in the sex organs, the testes and the ovary, to form gametes. We'll deal with the easier one first, mitosis. What are the consequences of mitosis? One cell divides to make two. Okay, that's easy enough to understand. Okay, but it's a diploid cell, which we represent with the term 2N. A diploid cell has two of each type of chromosome. You get 23 chromosomes from your mother in the egg and 23 chromosomes from your father in the sperm for a total of 46 chromosomes. The 23 chromosomes from your father match the 23 chromosomes from your mother. They don't match genetically. They match for what they code for genetically. For instance, if you have a birthmark, okay, the birthmark loci or locus will be on a certain spot on one of the chromosomes, maybe sometimes even more. It'll be on the same spot on the other parent's chromosome. Okay. Mitosis, the consequences of mitosis are that one diploid cell divides to form two diploid cells. If you have one, you have something. If you have two, you have twice as much. If you have 46 chromosomes in one diploid cell, you're going to have 46 chromosomes in each of the two resulting diploid cells. That's 92 chromosomes. How did you double the amount of DNA to make those chromosomes? The chromosomes themselves are able to divide. They are also able to produce more genetic information. We'll see that in just a second. This duplication in preparation for mitosis, straight cell division, this preparation of doubling the amount of DNA so that it can be split in half and each of the new cells have the proper amount. This happens in interphase. Interphase is the phase between the actual physical division of cells. When I was in school, they taught us wrong. They taught us that interphase was the resting phase. That's the farthest from the truth. In interphase, that's where the cell functions. That's when the liver cell processes and makes enzymes. Okay. It's in interphase that the DNA and the chromosomal material will double in preparation for division. It should never be called the resting stage. In humans, the nucleus of a normal somatic cell contains 23 pairs of chromosomes. Said that earlier. 23 pairs is 46 chromosomes. So that's the total number, 46, in a cell. Okay, so think of a cell. Okay, I like to think of the liver cell. You have 46 chromosomes in it while it's functioning in interphase. The ploidy number, something that people don't understand, but it's very simple, is 23. The ploidy number is N. The ploidy number is the number of types of chromosomes you have. In any one of your somatic cells, you have 23 chromosomes from your father. That's a complete set. That's the ploidy number. You have 23 chromosomes from your mother. That's the complete set. That's the ploidy number. When you got them together in the same cell, mother and father's chromosomes, it's 2N, diploid. The term N is actually pronounced haploid. If you have a deck of cards, you know the bicycle cards, there's a red deck and there's a blue deck, okay? Let's just take a blue deck out. Let's separate it into the 4 suits. Now, in plants, you can have 4 sets of chromosomes. We don't have that in humans, okay? But in each of the suits, spades, clubs, hearts or diamonds, in each of the suits, how many cards are there? I guess in this digital age, nobody plays cards anymore.
Is it 13?
13, right. Okay, not counting any jokers or anything like that. So the ploidy number in a deck of cards is 13. N equals 13. Okay, and they have 4 sets of chromosomes. Well, in humans, we have 23 types of chromosomes, and that is our ploidy number. Okay. The fact that your mother and father's chromosomes came together to form you meant that you were mixing genetic material, of course, and very likely to turn out different than your siblings or anybody else for that matter. Here are 2 chromosomes. In the center is a centromere. The centromere has also been called a kinetochore. I will see to it that I only use the term centromere, okay? And what we have here are 2 what we call homologous chromosomes. That means this might be chromosome A from your father. This is chromosome A from your mother, okay? And there's going to be 22 more pairs of these chromosomes, but we're not going to draw them all in for a total of 23 pairs. This has 1 strand of DNA. And the centromere is located somewhere along it. It doesn't have to be dead center. And the mother's chromosome has 1 strand of DNA. When they mature, when these mature, and they would look this way right after division, look what happens. The chromosome from the father has copied itself. The part on the right and the part on the left are identical copies. When you don't have anything else to copy, it's going to be identical. So gene A, same quality. Gene C, same quality. Do not look at these as crossing each other. Okay. Actually look at them as going straight down. Okay. So these chromatids now number 2. They're called sister chromatids. In the mother, that same chromosome also doubles its DNA, another strand of DNA, another strand of DNA. In some plants, again, there can be as many as 8 strands, but we're talking human anatomy, of course, and as far as we've ever seen, there's only been 2 strands of DNA. Over here, we had 2 chromosomes. How many chromosomes do we have over here?
4.
2.
Mm.
Okay, it's still 2, even though the chromosome has 2 chromatids.
Oh, right.
They are joined at the centromere.
Okay.
The number of chromosomes that you always have is the same number of centromeres that you have, no matter how many strands are joined to it. So here's 1, here's 2. Same over here, 1 and 2. So what happens then during interphase? While the cell is functioning and acting, it's also functioning to produce more DNA, getting ready to split and share this DNA to 2 cells instead of the one it's coming from. This is called replication. Here are the stages of mitosis. Okay. We looked at a cell the other day, and these stages and happenings refer to that previous drawing. In mitosis, somatic cell division, we start with prophase. The cell is diploid. The cell is 2N, okay, meaning 2 complete sets of chromosomes. Okay. It's ready to divide. The nucleolus, which produces transfer RNA, disappears. The chromatin that we talked about the other day, which was the dispersed DNA, soon condenses to become visible. What's happening is that these chromatids are twisting shut, tightening. You've all done this with a piece of rope or something. Okay. And as it tightens and looks like this, you have what we call a visible chromosome. In actual pre-prophase, in interphase, you would be able to see these guys because they would all be fibrous. Okay, so the nucleolus disappears, the chromatin condenses, soon to become visible chromosomes. The nuclear membrane itself begins to disintegrate. Books use the word disintegrate, fall apart, get consumed. None of these are really accurate. It's hard to say what happens to the nuclear membrane. I think it just spreads out it joins all the other membranes that it's attached to, meaning the RER and the SER. But we'll just go with the party line here. The nuclear membrane begins to disintegrate. And the centrioles, which we haven't talked about, we will, split in two and migrate towards the poles. These are the centrioles here. Textbooks always show one of them going to the other side. That's bull. They both move away from each other until they're opposite each other. Those are the centrioles. Okay, so I guess we'll come back to here. This is interphase. Interphase is the functioning cell. Everything is doing what it's supposed to do. The nucleus is doubling its DNA content. Okay. The cell is 2N. It has a set of chromosomes from each parent. The centrioles are getting ready to move, and that happens in prophase. Here the motion of the centriole is diagrammed. Okay. And the chromatin inside is condensing. And now it looks like those little X's that we just saw a moment ago, the chromosomes. In metaphase, the next phase, the centrioles will have reached their poles opposite each other. And they set up a spindle apparatus. That's this. These are fibers. These are fibers. Can you imagine yourself out in the ocean, deep sea, tuna fishing? You're on a boat. You throw out a line and you catch a tuna. This one throws out a line, catches the same tuna from the other side. Okay. So what's going to happen in anaphase is that the centromeres split, being pulled from both sides, and a chromosome piece of each homologous pair goes right and left, right and left, right and left, right, and they pull apart from each other. We have 4 chromosomes going to the left. Why do I call these chromosomes? Because you count the number of centromeres, and that is the number of chromosomes. However, if you even have a part of a centromere, if it's split in half here, then that's attached to a chromosome. So whereas there, in this example, 4 chromosomes prior to anaphase, at anaphase, the centromeres split and the information goes right and left. A full complement of chromosomes, but only one chromatid. Well, we know we can make more chromatids. A full ploidy number of chromosomes going both ways, right and left. Now, the chromatid or chromosome, as you wish, that I'm pointing to right now, and this one over here, right now, are identical genetically. There's no differences. Recombination doesn't occur here. Okay. What you did, you formed two chromatids on one chromosome. We did that way up here. Just to remind you where it was. And now this and this are identical images. So you're going to get a nucleus eventually over here and a nucleus over here that have exactly the same genetic information. And in a telophase, the cells furrow. Furrow is the term for breaking apart from each other. The nuclear membrane comes back. The chromosomes, although they only have one chromatid on it now, they'll disperse and become chromatin, and they'll form the other chromatid again, and this one again before they divide again. The centriole will regenerate itself so that there are two of them in each cell, as we started with. We end up in mitosis with two cells, genetically identical, each with a full complement of chromosomes. In humans, that's 46 and 46, each with the ploidy number of 23 chromosomes from the mother, 23 from the father, and they never mixed. They never mixed. They just duplicated. Now going up to the little part that I may have skipped here. Metaphase. The spindle apparatus has been set up. The chromosomes are aligned on the equator of the cell. Chromosomes do not need to pair off at this stage. It could occur, but they don't need to pair off. If you're thinking ahead to meiosis, not necessary here. The nuclear membrane has disintegrated by this time. Okay, in metaphase, the cell is still diploid. Here's metaphase. The centrioles have split, migrated, set up an apparatus, okay, and the chromosomes are roughly aligned on the equator of the cell. Most books call this the equator, but the equator goes around the circumference, and this is really a disc inside of the cell. So it looked like a nickel in shape. And all of these chromosomes would be floating on that. Once we leave metaphase, anaphase, the cell will split and temporarily will be 2n plus 2n. We'll come to that in just a second. The centromeres split, which doubles the number of chromosomes. Each chromosome resulting from the splitting of the centromere takes with it half of the chromatids. When the centromere splits, it's considered that the result is two centromeres, or two pieces of centromeres. The chromosomes migrate towards the centrioles. Okay. In anaphase, we have the splitting of the centromeres. Half of the centromeres and their associated chromosomes go to to each pole. You have 46 chromosomes going right, 46 chromosomes going left. That's 2n plus 2n. But it doesn't last in that situation for very long. Okay, you've got one cell that's got twice as much chromosomes, 92, but that quickly separates through furrowing. And then you'll end up with your original 46 and 46. Now, I think the first time through can be a little bit difficult when you're using somebody else's drawings that you haven't seen before. There are so many depictions of this on the internet. Each one takes a little special look. So if you do go on the internet, you see this or a version of this, realize it's probably accurate, very accurate, just being demonstrated in a different way. Okay? Okay.
Charlie, at the very end, when you said each one ends up with 46 chromosomes, were you referring to telophase, not cytokinesis or cytokinesis?
Actually, during telophase, things aren't separated completely yet.
Okay,
Okay.
thank you.
But you know, I wouldn't be so picky as to even ask you that.
Okay.
Here, cytokinesis, the separation of the cell. Okay. Yes, then you've got back to 2n and 2n.
Okay.
They're separate.
Thank you.
Okay. Meiosis. Okay, I think it's probably easier to go to the drawing and then come back to the text. Okay. Here we can start with this. Come on. Meiosis is only for the production of gametes. A gamete has to have half the number of chromosomes as an embryonic cell, as the embryo itself. This results from a series of two divisions of cells that are going to produce gametes. Now, where are these cells? Specifically, in the seminiferous tubules of the testes. The cells there are called spermatogonia. I don't have that word on here. I just want you to understand where we're talking about. In the female, the oogonia, that word you won't find here either. If it's not here, I won't ask it. But the oogonia undergo a series of two divisions to produce up to 4 cells. Okay. The 4 cells that result from the two divisions of the spermatogonia are all spermatids. They're not fully mature sperm cells. They don't have a tail. They don't have a neck or a collar. They don't have an acrosome, which is an enzymatic pouch at their tip to burst through the membrane of the ovum. They're not ready yet. But the two divisions resort result in the formation of 4 haploid cells. In the female, the division of one large oogonium results not in 2 equal cells. It results in one large cell and a little tiny cell. And the little tiny cell is going to die. It'll get reabsorbed, can't do anything. And then the one large cell divides again. And it'll divide unequally and form one more large cell and another small cell. These small cells are called polar bodies. We see them, we know they're there, but they're useless. The idea in the female is to undergo meiosis and leave one large cell that's got food inside of it. In comparison, the sperm cells that essentially attack the ovum, they are minute. All they bring with them is DNA. You know, no dowry, no gift, no nothing. Okay. Why do we have to go through meiosis? Whether it's in the male or the female, you need to go through meiosis to form the haploid gametes, and the gametes will be recombined, meaning that they will have different combinations of genetic material. Gametogenesis is the formation of gametes or sex cells. Spermatogenesis is gametogenesis in the male. It's the formation of sperm cells, and it goes on in men from puberty until death. In the female, the production of ova takes place in the ovary at birth. It does not continue on. At birth or there a few days either side, all of the eggs that are going to be in a particular human female will have been formed. If they're used up, there aren't any more. Okay. So these are the 2 phases of mitosis here, side by side, 2 divisions of meiosis, I should say, not mitosis, meiosis. We really need to concentrate on the first one, but think about what is the function of meiosis. The function of meiosis is to produce gametes. The function of meiosis in producing gametes is to produce gametes that have different genetic combinations so that we get variability. We start at interphase. All of the genetic material is reproduced in interphase, just as it was for mitosis. So the doubling of the chromatids, just like it did in interphase, as it did in mitosis, is done in interphase. At prophase, nothing much different here. The centrioles migrate. But now the chromatids despite this drawing, are not fully tightened, and the chromatids will line up chromosome for chromosome. Chromosome A, mother A. Chromosome B, mother B. They match up next to each other. And the chromatids cross over each other, entangle, pull apart. Okay, this doesn't shred anything. It exchanges the tip of one chromatid with the tip of another chromatid. This is happening in 23 places in the cell because you have 23 pairs of chromosomes exchanging their information back and forth, back and forth. So back and forth, does that mean you take it and then give it back? No. You're shuffling a deck of cards here. And you are really shuffling the genes that line the chromatids. The chromatid on one side of a chromosome will no longer match the chromatid on the other side of the same chromosome because it has exchanged parts with the analogous chromosome from the other parent. Now you're mixing mom and dad's genetic information. That's the main thing you have to get, is that the mixing or recombination occurs in meiosis number 1. Synapses, that's crossing over, and crossing over, saying it again, occur here. It's in prophase. Now, the chromosomes will completely condense. They'll be hooked by the spindle fibers from the centrioles. Although they are different color down here, one doesn't come from the father, one from the mother. It's just to show that they are different. These have exchanged their genetic information so many times until the deck shuffling comes to a stop. Same thing up at the top here. And now when you pull them apart, the centromeres don't split. The centromeres, as you recall, split in mitosis. Instead, the homologous pairs separate. That just means the pairs of chromosomes. One goes one way, one goes the other. One goes one way, one goes the other. Why all these different colors? Because they all are genetically different, not just chromosome to chromosome, but chromosome to homologous chromosome, the one from the other parent. If we had 4 chromosomes up here, we only have 2 being pulled in each direction down here. So the number of chromosomes is halved. And once the splitting continues, each of these cells is haploid. 4 chromosomes become 2. 46 become 23. And 23 end up in each of these cells. Recombination, that's shuffling here. Recombination happens in meiosis 1, but the reduction down to one end, haploid, also occurs in meiosis one. Okay. That's going to be a question somewhere in your future. Okay. Recombination and reduction occur in meiosis one. Meiosis two is quick. It's just the taking of these two cells that meiosis one formed. They're already haploid. Okay. Each chromosome is containing at least two chromatids. Okay. The centromeres do not split. That's the reason this all occurred. In meiosis 2, we go from here up to prophase, set up metaphase. I'm not going to say anything about it. Now we will pull the chromosomes apart, breaking the centromere. In breaking the centromere, we'll have one end's worth of chromosomes going one way, one end going the other. Why isn't it two in and two end? Because we started with one end down here. One end, one end, one end, one end, one end, one end. And now we separate them and break the centromeres. Okay. Two, 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 ending up down here. The four cells that result, because this breaks to two, this breaks to two, this separates, this separates. The four cells that result are haploid, and these are going to serve as the spermatids. These are not going to serve as the egg and the polar body because they're all the same size. And as I said a moment ago, the female will have one ovum and two, sometimes three polar bodies, which are useless. The same thing happens in oogenesis, the formation of ova, as happens in spermatogenesis. When we're talking about the nuclear material, it doesn't matter that one cell breaks into a big and a little cell. The chromosomes do not take up much space. We just
I have a
set up, yes.
For anaphase 1, the centromeres do not split, but anaphase 2, they do split.
Yes.
Okay.
Yes. So the failure to split anaphase 1 is how we can reduce the number of chromosomes in the two cells that are going to result. Well, now that we've talked about that, you can set it aside because it's not going to be on Wednesday, Tuesday's test. Okay, and time 3:32. Okay, let's come back at 4:45. Okay, I'm going to run a little longer today. I'm sorry, but I'm going to show you more bones in review. I'm going to use bone models rather than diagrams. Okay, so see you at 4:45. Okay. Okay, any comments? Well, let me hear somebody say something.
We're here. We're on.
All good.
Okay,
Yeah, we're good.
can't tell. Okay, I'm going to go over some bones. It seems the correct thing to do, since the bone practicum is next week. And first, I'm going to show you the range of material that you'll have. And then we'll come back here.
Professor, quick question.
Sure.
It should be expected that I should know the bones as they are outside of the colorful pictures, correct?
Yes.
Okay, thank you.
Yeah. There are no colorful pictures on the exam, actually. And don't worry about that. I mean, I'm not worried because if I were going to get a lot of complaints, that would worry me, but I don't. Okay. I'm going to have to turn off the recording for a second, and I don't know if I can do that here. Damn it. Okay. Come back. I'm going to pause the recording. Okay, so now we're back to it. So there'll be a little gap there. I told you why. Now, okay. What you need to do, if you have it already, is to put this into speaker view. Okay. Are you looking at a whole bunch of tiles? Who is?
Yes.
Okay. And who sees just me?
I see just you.
Okay. Now we got to find where the speaker under views. I'm here at the top left, no, top right of the black area. It says view. Okay, I'm at gallery right now. I'm going to click speaker. Okay. Okay, so now you should be able to see me. The idea is here, you can't see the bones otherwise. Okay, not a little. No, I don't even want that. But it's there. Okay, and now you're going to have to bear with me and help me out to set the bones. Toughest one to do for me is going to be the skull. And I know from past experience that it will be tough. Too much light. Okay, the other bones will be a lot better. So I'm only going to do what I can do on this. Okay. The point down here, that's the styloid process we talked about. Here's the mastoid process right behind it. And all of that is a part of the temporal bone. Let me see what's best. Here's the coronal suture. That's the trouble with white bones like this. You get no contrast. I'm okay. I guess I got to keep talking. If I don't keep talking, then somebody else pops up, huh? Zygomatic arch, zygomatic bone. The maxilla. Now you can see the partition or separation. The lower portion here is the vomer. The upper portion is the perpendicular plate. Look inside of the eye. Oh, I wish I had somebody to move a camera for me. There we go. This is superior orbital fissure. The inferior orbital fissure. I'm purposely trying to pick out things that I think you might have a little bit of a problem with. And in the back, that little hole is the optic foramen. At the front and embedded in the frontal bone. This is the ethmoid bone, the raised part, cristagalli. On either side, the cribriform plate, here and here. The cribriform plate has holes through it. They're called olfactory foramina or cribriform foramina. The large hole at the bottom, of course, is the foramen magnum. And you're looking at the posterior part of the skull right here. So here is the jugular foramen. This one is probably the toughest on the test. This is the internal auditory meatus, right above the jugular foramen. This hole is the foramen ovale, right behind it. Foramen spinosum. Foramen rotundum is up here. And here, the foramen lacerum. This is sphenoid bone, and it's the lesser wing of the sphenoid bone. Here's the sella turcica of the sphenoid bone. The greater wing is down here, here, over here. That's the one that has the many holes, foramen spinosum, ovale, and rotundum. Although you can't see it here, I'm going to touch it anyway. Right there is the sphenoid bone on the outer side. And in the eye, here is the sphenoid bone. My fingers covering it, fingers off of it, covering sphenoid bone. Maxilla. And I can't show you the other ethmoid in here. It's just not possible. Here's the inferior or infraorbital foramen right here. The supraorbital notch is barely visible right here. Okay. On the underside of the skull, can't forget the occipital bone, the occipital condyle, the occipital condyles right here. And on the posterior side, down here, separating the nasal cavity in half vertically. That's all vomer. This is the vomer. What happened to the perpendicular plate? It doesn't come back this far. Here are the pterygoid processes. Here. Here. Here's the palatine process of the maxilla. And at the end, at the back here and across, is the palatine bone. Does not show well on this model. Okay, that's the hardest one to do. It's just crazy. The first time I did it, I was much worse. I was bad enough right now. You want to point left and you go right. It's just a mirror image of what you want to see. Plus, on the skull, I know this bleached white skull does not show sutures and stuff very well, unfortunately. But I wanted to show you some of the structures that I could on a real, not a real skeleton, but a plastic model anyway. Is there another part of the material that you would like to go over? Well, think about it and let me know in just a second. If for you, the best way to go over this material is whatever way you've already developed, then you don't need to be here. I mean, it's fine to exit at any point. But if you think that you'll miss out on something that, or miss out on being able to ask a question or see a specific structure, and I can pull it out of the bone box, which is behind me, then you should stay. So if you're going to stay, you should have hopefully some questions. And if you're going to exit, I'll see you on, no, I won't see you on Tuesday, but I'll know that you're here. Okay. You won't see me, but I will be at the test all of the time. Okay. Okay. So either take your leave or let's go on with some questions.
Could you go over the foramens again, like the foramen rotundum, ovale, and spinosum?
Okay. That one I just did, but want to see which angle is best. Okay. Where's my little pointer? Obviously, this is the front. Okay. I've got the guy looking at me. The sphenoid bone runs across here, all the way across here, and to the other side. And in the greater wing of the sphenoid bone, which is this lower part, okay, this is where we find the material you're asking for. This is a foramen spinosum. Looks like the spine of a high heel shoe to me. The foot of the high heel shoe is the foramen ovale, right there. The foramen rotundum is right there. I always think of it myself as a foot kicking a ball away from it. So spinosum, ovale, and rotundum here. The lacerum, which is usually asked about, is right next to it. Right there. Foramen lacerum.
And then where was the internal auditory meatus again?
Okay. That we go back to the jugular. Now let's use this one. There's the jugular foramen. And above it, this is the internal auditory meatus or canal. Let's look on the other side. It actually shows better. Jugular, internal auditory meatus up here.
All right, thank you.
Thank you. I just figured out how this thing works. If you say something like Harrison just did, and nobody follows you up with anything but silence, then Harrison is on screen all the time until the next person talks. I'll be on screen now until the next person talks.
Charlie?
Yeah.
So what we learned today with the knee, is that also going to be on the test?
Yes.
Okay, thank you.
Charlie, was the stuff that we learned today about the mitosis, is that going to be on the practical too?
No.
Okay.
No,
Thank you.
none of the lecture material is going to be on the practicum.
Okay.
You only need to study the lab material.
Okay. Just double checking. Thank you.
No, that's fine. I'll say something so we get you off the screen.
Wait, so sorry, last question. Where was the carotid foramen or canal?
Okay. The carotid cannot be seen from the inside. So we have to go over to this side. And it's this one. No, it's not. I'm not seeing it right. Okay. Here's the jugular again. Okay, the large one. Right above it is the carotid canal. The carotid canal I'm doing it on the other side. Okay, the one that's going through in Stark, that's the jugular. It's going backwards. Goes all the way through. Right, there's the carotid is right above it. The carotid does not go all the way through. It makes a turn and goes through this bone and comes out over here at the foramen lacerum. Let me see if I can get one better angle on this. got to be closer, obviously. Don't let me make you dizzy here.
Charlie?
No, not yet. Let me finish the question. Then I get to you. No, just make any noise. You take over the screen. I can't answer this question. Okay. This is the carotid canal here. And on the other side, it's here. Right next to it is the jugular. And the jugular. Okay, Mai.
Where is the optic canal?
That's in the eye. And I can push this through. No, I can't because it's too big. See the round hole at the very back? Dark
Oh,
little round hole.
okay, I see.
That was
Okay,
it. Yeah.
okay, thank you.
Okay.
I am confused on the coronoid process and conoid. I saw it in the clavicle and the elbow and the.
No, you saw the coronoid process
Mhm.
in the ulna. Okay. Elbow is not the answer to anything. It's not on your sheet. But it's the
Coronoid.
coronoid
Process.
process of the ulna. Yes.
Mhm.
The coronoid process of the jaw, mandible.
Oh, okay.
Okay. And you didn't see any coronoid process on the, you said clavicle? No, there's none. There's a conoid tubercle.
Scapula? Scapula?
Conoid tubercle. And there's a coronoid fossa. Coronoid fossa that the ulna fits into when you flex your arm up. It's on the heart side. On the heart side. So you need to look that one up when you go through these. See if I get lucky.
Mhm.
Okay. On this bone, this is the humerus, upper arm bone, and head. Neck. Come on down to the distal end. Okay. This rounded bump is the
Capitulum.
capitulum. Okay. You have to be, sorry, but you have to be quiet while I'm explaining it. Otherwise it takes my picture away. I don't know if it takes it away from you, but I can't see what I'm doing.
Okay.
Okay. So here's the capitulum. No, okay. Here's the capitulum. Here's the trochlea. The radius rotates around and into this little groove here where my fingertip is right now. That's the radial fossa. The trochlea, okay, going around it, the coronoid process of the ulna fits into the coronoid fossa. That's the coronoid fossa. So you have a radial fossa, which is easy to find above the round ball. And then the coronoid fossa. On the medial side, this is your medial epicondyle. Medial epicondyle right there.
Thank you.
Thank you.
Charlie, I have another question.
Sure.
How many orbit bones? 6 or 7?
There are in reality 7, but only 6 that I care that you know. I don't care about you knowing the palatine bone because it's so small. You know, I couldn't show it to you to make you believe it. So you're looking at frontal bone, zygomatic bone, the
Isthmoid,
maxilla, ethmoid, lacrimal, and frontal.
Okay. Thank you.
Charlie, which one did you just say was so small?
Palatine.
Thank you. Is the maxilla easily confused with the alveolar process?
Not the way we label it.
Okay.
No. I think the only person that's ever complained or been dissatisfied with the labeling of a question has been me, and I changed that right away. I haven't had any complaints in years and years. It's not always the same test, but still, I'm glad not to get any complaints.
The pictures, you may have already answered this. The pictures on the exam, are they pictures that have been built into our modules?
No.
Okay.
No. If that were the case, you'd just be learning the pictures.
Hmm.
Don't worry about it. Honestly, don't worry about it.
Charlie?
Yeah.
Do you want to submit the chapter 1 and 6 separate or together?
Whichever way is easiest for you.
Okay. Thank you.
Is the zygomatic arch the same as the zygomatic process?
No,
No.
Okay.
but I'm telling you, you don't have to know the difference. So if I explain it, I'm going to confuse somebody. That's what I'm afraid of.
That's okay. I was just going through my list and I was going to all the different figures and I found zygomatic arch, but well, yeah, I was confused. Okay, but that's okay.
Okay, well, just think of it this way. The zygomatic arch is composed of two bones, a long piece and a short piece.
Okay.
And don't worry about anything further.
Okay.
Okay, I mean, I can explain it, and I used to, but it just drove people crazy.
Okay. Thank you.
I have a question regarding
Yep.
the pelvis. Is the iliac fossa only on the anterior view? And then the ilium is on the posterior?
The iliac fossa is a part of the ilium. Okay, so you
Okay.
can't separate them, except to say that the iliac fossa is only visible, as you said, from the anterior.
Okay, thank you.
Okay. When you guys finish talking, I'll say something just to take your picture off of the screen. Jessica, what were you seeing on the screen? Yourself?
No, I was seeing you.
And what are you seeing right now?
You, I have you pinned, so it only shows your point of view. Or
Okay. 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 I was just saying though that I'll say something in case we're leaving somebody up there doesn't want to be seen, but we'll find out.
Charlie, sorry, I didn't get it. We will use the conference Zoom on the practical one.
Nope.
Okay.
Go to the mod. I'm going to send the thing out over the weekend.
Okay.
Just to be sure that everybody knows. A couple people weren't here today, and I want to be sure that they know as well.
Okay, and I think you said that you will send a list of something.
Oh, for 3 or 4 things that got added. Yes.
Okay. Thank you.
Not hard.
Okay. Thank you. Last question. Sorry. The grades for quiz practice will add to our grades or not?
No.
No.
Not doing the quiz is going to hurt your grade, in my opinion. Okay. So I have no problem with letting you take the quiz or not take the quiz. I want you to take the quiz because you should want to get the best grade you can. Okay? It only takes a couple minutes anyway. There are only 20 questions. Okay.
thank you.
Bye-bye.
Thank you, Charlie.
Thank you,
Goodbye.
Charlie.
Thank you, Charlie.
Good luck, everybody.
Bye-bye.
Thank you.
Take it easy.
So we won't see you till Thursday, right? Your face?
Yeah, yeah,
Okay. All right. Thanks. Have a good weekend.
you too.
Thank you, Charlie. Bye bye.
Bye-bye.